Hi, my name is Josh Currie, and I'm a new assistant professor in the biology department. I'm really glad that you could join us for our recruitment week here at Wake Forest. And I'd just like to introduce myself and tell you a little bit about the research that we do in my lab. So, my lab is just starting, and we're recruiting master's and PhD students to start in the fall and kind of start to build our research program here at Wake Forest. So, our main research question in my lab centers around the idea of extreme regeneration. So, we know that in humans and a lot of other mammals, the ability to regenerate is diminished or even gone completely. And we would love for that not to be the case because that would have a huge positive impact on human health if we could regenerate our tissues, our organs, and heal without forming scars. And we know that certain animals across the animal kingdom have retained or have an exceptional regenerative ability. So we'd like to understand what separates and differentiates those two different outcomes. And in particular, we're interested in the regenerative structure. So if we look at a salamander, which is able to regenerate its limb, what happens after the limb is amputated is that cells start to accumulate at the wound site and form this mass of cells that is called the blastema. And this blastema will go on to recapitulate all the missing structures and tissues that were taken away in an amputation. And the animal model that we actually use to answer this question is the Mexican axolotl. It's a classic amphibian model that people have used for over 150 years. And this animal really is a super exceptional regenerator. It can regenerate parts of its brain, its heart, its lower jaw, its gills, its lungs. It can regenerate its entire tail, which is incredible considering that the tail also includes the spinal cord, so it can recover its central nervous system. And in particular, my lab primarily studies regeneration in the limbs because we know a lot about the process, and yet there's still a lot that we don't know. And the way in which we do this in my lab is really focused on watching cells and molecules and trying to understand how, on a cellular and molecular level, this process happens. And we do this in a couple different ways that are primarily focused around microscopy, although we're venturing out into other techniques as well. But this is primarily how we look at regeneration. One, using a genetic trick where we can label cells with unique colors and then follow them and their progeny over time. So we have this multicolor labeling that we call limbo, and we follow cells throughout the time course of regeneration that can take days and weeks. The second way that we look at regeneration is by following cells in a very fast manner, so acquiring images every few minutes so we can see what individual cells do during the process of regeneration. So here you can see immune cells that are moving around, clearing debris, and assisting in the process of regeneration. Lastly, one of the things that we do is we often take cells out of the animal and put them in a dish. And in a dish, we have a much more controlled environment where we can look at particular processes and molecules that coordinate regeneration. So here you can see some cells migrating in a dish. So some of the big questions that we have in the lab, and this is just kind of a sampling of some of the things that we're really interested in, is, what are the signals that activate and coordinate regenerative cells? How are they perceived? Where are they coming from in the environment of regeneration? And how are they used to choreograph this entire process? Secondly, one of the things that we know is that cells that are in that injured tissue, they somehow have to go back in time. So they are reprogrammed to an earlier, more naive state so that then they can pattern into all types of different tissues that they need to become, such as cartilage and tendons and all the soft tissues. So how do they do that on a genomic level, on a cell and molecular level? And finally, we really want to understand the environment that facilitates regeneration, as opposed to in humans or other mammals that facilitates scar formation. So what is the microenvironment or the niche that cells find themselves in during regeneration, and can we identify that and perhaps even replicate it or reconstitute it? So that's just a flavor of what we do, and I'm really excited to talk about projects with individual folks that are interested. So what I would also encourage you to do is to check out the lab website, which I've listed here, and which you can either go to the address or you can find it from my faculty page at the Department of Biology at Wake Forest. And what I would specifically encourage you to look at are the research and resources pages, which will tell you a little bit more about regeneration in general, and also questions that we are looking at in the lab, and maybe even provoke some ideas that you might have for a thesis project. Then I would also encourage you to look at the core values and expectations page, which tells you a little bit about the culture that we're trying to build in the lab and what the mentorship and ideas that we're trying to facilitate for training folks, and to make it just a great place to do science every day. So again, I'm really excited that you're here. I hope that I get a chance to talk to you, and welcome to Wake Forest.